Welcome back to the deep dive. Today we are embarking on, well, a really ambitious journey.
Definitely ambitious.
We're cracking open the source material on the mammalian cell, its fundamental structure, molecular workings, the whole package. Our aim isn't just a surface skim. We're going for genuine, professional-level mastery here.
Right. It's like getting the architect's blueprints and the engineer's notes all at once. To really get cell biology, you absolutely have to appreciate the incredible detail.
From the overall shape down to the tiniest protein machines,
Right, exactly. And the signaling pathways, I mean, they literally decide life or death for the cell. The core idea we need to keep coming back to is how structure and function are just, perfectly intertwined, down to basic physics even.
Okay, let's start right there. Basic physics and general characteristics. The constraints on cell size and shape that jumps out immediately.
Yeah.
Why are most mammalian cells so tiny, like 5 to 50 micrometers?
It really boils down to one hard limit, the rate of diffusion. Simple physics. The cell needs to constantly bring in fuel, oxygen, building blocks, and get rid of waste products. Diffusion across the membrane and through the cytoplasm has to keep up with the demands of the entire cell volume.
Ah, the surface area to volume ratio problem.
Precisely. As a cell gets bigger, its volume increases much faster than its surface area. So if it gets too big, the center just can't be supplied quickly enough. It starves or drowns in its own waste, effectively.
Which explains why, say, a lymphocyte is maybe 6 micrometers and a red blood cell around 7.5. They're limited by that supply line.
That diffusion boundary sets the fundamental scale for most cells. But like any good engineer,
The cell finds workarounds, ways to cheat the system.
absolutely. If a sphere isn't efficient enough, change the shape. Neurons are the classic example. They can be over a meter long.
How do they manage diffusion then?
By being incredibly long and thin. The axon maintains a good local surface area to volume ratio all along its length. And, crucially, they rely on very sophisticated internal transport systems, molecular motors trucking cargo up and down the axon to cover those huge distances. Diffusion alone wouldn't cut it.
Okay, so shape is one strategy. What about just being giant, like megakaryocytes or osteoclasts? They can be over 200 micrometers.
Right, those are exceptions too. They often achieve that size through things like having multiple nuclei, or by fusing multiple cells together. They become specialized factories, megakaryocytes for platelets, osteoclasts for breaking down bone. Their sheer size serves a specific, massive function.
And that idea of structure serving function applies just as much to the cell surface, especially for transport. We see incredible folding in absorptive cells.
Oh, absolutely. If your main job is moving stuff across the membrane, think gut lining cells absorbing nutrients, you need to maximize that border cross.
Or surface area.
Exactly. So you see intense folding, apical microvilli, these finger-like projections sticking out into the lumen, and basolateral infoldings on the other side. It dramatically increases the real estate available for transport proteins and diffusion.
Before we move off general characteristics, let's touch on motility. The source material calls it a universal trait, but it's more than just cells crawling around, isn't it?
Much more. We tend to picture a macrophage chasing a bacterium, extending pseudopods or lamellipodia,
Yeah, the classic amoeboid movement.
and that's definitely part of it. But motility, in this context, covers all movement. The constant stirring and streaming of the cytoplasm itself.
Internal currents.
Right. The beating of cilia, like in your respiratory tract moving mucus, or the flagella whipping on sperm for propulsion, even muscle contraction, that's specialized cellular motility, and the intricate dance of chromosomes during cell division, it's all under the umbrella of motility. The cell is fundamentally dynamic, never static.
A dynamic entity needs a boundary that's both flexible and controlled. Perfect segue to section 2, the plasma membrane, the cell's border.
The border, the gatekeeper, the communication hub, it does a lot. We still fundamentally use the fluid mosaic model, which holds up remarkably well.
So lipids and proteins in roughly equal measure.
Yep. A sea of lipids, primarily phospholipids, cholesterol, glycolipids, forming that core bilayer structure. Hydrophobic tails tucked inside. Hydrophilic heads facing the watery environments inside and out. And floating within or anchored to this lipid sea are the proteins.
Crucial point. It's fluid, but not just a random soup.
Exactly. Fluidity is key for movement and function, but it's regulated. And the proteins aren't just scattered randomly. Let's talk protein types first, though.
Okay, the big players.
You've got your transmembrane proteins. These guys span the entire width of the membrane. Often they're large complexes, forming channels for ions, pumps for active transport, or receptors to receive signals from outside. They're the real workhorses of membrane function.
And the others.
Extrinsic proteins. These are more loosely associated, usually bound to the inner cytoplasmic face of the membrane, often by linking up with transmembrane proteins or the cytoskeleton just beneath the membrane. They play roles in anchoring signaling relays.
The source material mentions freeze fracture techniques showing particles mostly on one face.
Ah, yes, the intermembranous particles seen in freeze fracture. These often represent those large transmembrane protein complexes. And the key finding is they tend to stick predominantly to the protoplasmic face, PF, the inner leaflet, the one facing the cytoplasm.
Asymmetry.
It reflects the different jobs the inner and outer faces have. The inside needs to connect to the cell's internal machinery, the cytoskeleton, signaling cascades. The outside is focused on receiving signals and interacting with the external world. So the protein distribution reflects that functional division.
Okay, so the membrane is fluid, but you mentioned it's not just a random soup.
Yeah.
How does the cell create specialized zones within this fluid environment?
That's where lipid microdomains, or lipid rafts, come in. Think of them as specific neighborhoods within the membrane.
Okay.
These are areas enriched in certain lipids, particularly sphingomyelin and cholesterol. These lipids pack together more tightly, creating regions that are thicker, less fluid, and more ordered than the surrounding membrane. They're even resistant to certain detergents in the lab, which is how they were initially identified.
And the functional significance.
Well,
Why bother creating these RAF?
efficiency and organization.
Right.
Imagine you need 5 different proteins to work together for a specific signaling event, like T-cell activation.
Right.
Instead of waiting for them to randomly bump into each other in the vast expanse of the membrane, the cell preassembles them onto a lipid raft. It's like putting all the necessary tools on one workbench. It hugely speeds up signaling, helps in protein sorting for transport, and is involved in membrane budding and fusion.
So specialized platforms for specific tasks. Clever.
Very. It adds another layer of complexity beyond the basic fluid mosaic idea.
Okay, that's the lipid organization. What about the outside, the fuzzy coat?
The glycocalyx. That's the carbohydrate-rich layer on the outer surface of the plasma membrane. It's formed by the sugar chains attached to membrane proteins, glycoproteins, and lipids, glycolipids.
Like a sugar coating.
Essentially, yes. And it varies a lot between cell types. It acts as a physical buffer, protects the cell surface, but also plays critical roles in cell recognition.
Recognition. How so?
The specific patterns of sugars in the glycocalyx act like molecular ID badges. This is fundamental for tissue integrity cells recognizing their neighbors. It's also the basis for things like blood groups, and hugely important in immune recognition and organ transplant compatibility. Your immune system checks the glycocalyx ID to see if a cell belongs or not.
And in some cells, it even has enzymatic functions.
Yes, a great example is in the intestinal lining cells, the enterocytes. Their glycocalyx actually contains digestive enzymes positioned right at the surface where nutrients are being absorbed. It's part of the digestive machinery itself.
Fascinating. Okay, let's move to the membrane's core function. Transport. Getting things across the border. 3 main ways, right? Starting with an easy one.
Passive diffusion. This is for molecules that can just slip right through the lipid bilayer itself. They need to be small, generally uncharged, and lipid-soluble.
Like oxygen, carbon dioxide.
Exactly. O2, CO2, steroid hormones, things that can dissolve in the hydrophobic core of the membrane. They just move down their concentration gradient from high concentration to low. No help needed, no energy spent.
Simple. But most things the cell needs aren't like that. Sugars, ions, amino acids, they're polar or charged. They can't just dissolve in lipids.
Right. They need help. That brings us to facilitated diffusion. Still passive, meaning it doesn't cost the cell direct energy. Movement is still down the concentration gradient, but it requires a protein helper embedded in the membrane.
2 types of helpers.
2 main types. First, carrier proteins. These bind to a specific molecule, like glucose or an amino acid, and then undergo a shape change, like a revolving door, to shuttle the molecule across the membrane.
1 molecule, or maybe a few at a time.
Yes, they have specific binding sites. The second type is channel proteins, often called ion channels. These form pores or gates through the membrane. When open, they allow specific ions like sodium, potassium, calcium, chloride to flow through very rapidly.
Much faster than carriers.
Orders of magnitude faster. But they are highly selective. A sodium channel only lets sodium through. A potassium channel only potassium, and so on.
And what controls whether these gates are open or closed? They can't be open all the time.
Absolutely not. They're tightly regulated. Some are ligand-gated channels. They open when a specific signaling molecule, a ligand, binds to the channel protein itself. Think neurotransmitters opening channels at a synapse.
Okay.
Others are voltage-gated channels. Their opening and closing is triggered by changes in the electrical potential, the voltage difference across the membrane. These are absolutely fundamental for generating nerve impulses and muscle contraction.
So passive and facilitated diffusion handle movement down the gradient. But cells need to move things against the gradient too, right? To concentrate things inside or pump things out.
Exactly. And that requires energy. This is active transport. It's the only way to move solutes uphill against their electrochemical gradient. The cell has to pay for this, usually by hydrolyzing ATP.
Why is this so vital?
To maintain the cell's internal environment, which is often radically different from the outside, think about nerve cells. They need a high concentration of potassium inside and a high concentration of sodium outside. That doesn't happen passively. It requires constant active pumping.
And the pumps themselves, how are they classified?
Based on what they move and in which direction, a uniporter moves just one type of molecule across, though this term can also apply to some facilitated diffusion carriers.
Makes sense.
Then you have the coupled transporters, which move multiple things. A symporter or co-transporter moves two different molecules in the same direction across the membrane. Often, it uses the downhill movement of one molecule, like sodium flowing in, to power the uphill movement of another, like glucose being pulled in against its gradient.
Using one gradient to build another.
Precisely. And the opposite is the antiporter. This moves two different molecules in opposite directions. The absolute classic example found in nearly all animal cells is the sodium-potassium pump.
The Na plus K plus ATPase.
That's the one. It pumps 3 sodium ions out of the cell and 2 potassium ions in, using the energy from one ATP molecule for each cycle. This pump is critical for maintaining membrane potential, controlling cell volume, and driving secondary active transport of other solutes. It consumes a huge fraction of a resting cell's energy budget.
Okay, that nails down the border and its traffic control. Let's step inside Section 3. The internal compartments, the organelles. The cytoplasm itself. It's not just empty space, is it?
Not at all. The cytoplasm includes everything within the plasma membrane but outside the nucleus. It consists of the cytosol, the thick, gel-like fluid itself, packed with proteins, metabolites, ions, and everything suspended within that cytosol.
Which includes?
Well, you've got the extensive networks of internal membranes, the cytomembranes, that's the ER and Golgi mainly. Then the distinct membrane-enclosed organelles, like lysosomes, peroxisomes, mitochondria. And finally, various non-membrane-bound inclusions, which might be storage granules like glycogen or pigment accumulations like lipofusin.
Let's tackle the biggest membrane network first, the endoplasmic reticulum, the ER. It's vast.
It really is. It's a continuous network of interconnected sacs, cisternae, tubules, and vesicles that extends throughout much of the cytoplasm. Its membrane is actually continuous with the outer membrane of the nuclear envelope.
And its key feature is creating separate compartments.
Yes, its membrane divides the intracellular space into two distinct environments. The cytosol is one, and the space inside the ER network, the intramembranous or cisternal space, is the other. This separation is crucial for its functions.
And those functions differ dramatically based on whether it has ribosomes attached, right? Rough versus smooth ER?
Exactly. The rough ER, our ER, looks rough because its cytosolic surface is studded with ribosomes actively synthesizing proteins.
Proteins destined for export or other organelles.
Precisely. Proteins that will be secreted from the cell inserted into the plasma membrane, like receptors or channels, or delivered to the Golgi lysosomes or endosomes. They all begin their journey by being threaded into the RER lumen or membrane as they're synthesized. The RER is also where these proteins start to fold correctly and get their initial sugar modifications, like acylation.
And it's got a built-in quality control system?
Absolutely critical. The ER-associated degradation, ERD system. The RER is constantly checking if newly made proteins are folding correctly. If a protein is misfolded, ER machinery recognizes it, pulls it back out into the cytosol, tags it with ubiquitin, and sends it off to the pretysome for destruction.
To prevent toxic buildup of bad proteins.
Exactly. It's a crucial checkpoint. If ERAD gets overwhelmed, it triggers what's called the unfolded protein response, a major cellular stress signal.
Okay, now the smooth ER, SER, no ribosomes, so different jobs.
Totally different skill set. Its functions vary a lot depending on the cell type, but key roles include lipid synthesis, making phospholipids, cholesterol, and steroid hormones. In liver cells, hepatocytes, the SER is packed with enzymes for detoxification, like the cytochrome P450 family that breaks down drugs and toxins.
Also involved in glucose metabolism in the liver.
Yes, it contains glucose 6-phosphatase, the enzyme needed to release free glucose from glycogen stores into the bloodstream, and a highly specialized form of SER exists in muscle cells.
Or cytoplasmic reticulum.
Right. The sarcoplasmic reticulum is basically a calcium storage tank. Its main job is to soak up calcium ions from the cytosol and then release them rapidly on demand to trigger muscle contraction.
We keep mentioning ribosomes. Let's quickly recap their role as the protein factories.
Ribosomes are these incredible molecular machines, about 25 nanometers across, made of ribosomal RNA and proteins. They have two subunits, the large 60S and small 40S. These subunits are actually assembled in the nucleolus, inside the nucleus, and then exported to the cytoplasm.
And they read the messenger RNA, mRNA code.
Yes, the mRNA carries the genetic instructions from DNA. The ribosome clamps onto the mRNA and reads the code, codon by codon, recruiting the corresponding amino acids carried by transfer RNAs and linking them together in the correct sequence to build a polypeptide chain of protein.
Often, many ribosomes work on the same mRNA at once.
Very often. That structure is called a polyribosome or polysome. It allows the cell to produce many copies of the same protein very quickly from a single mRNA template.
And the decision RDR bound or free in the cytosol depends on the protein's destination, encoded in its sequence.
Exactly. Proteins destined for the ER pathway have a specific signal sequence at their beginning. As soon as this sequence emerges from a free ribosome, it's recognized by a signal recognition particle, which escorts the whole ribosome mRNA complex to the RER membrane. The protein is then threaded through a channel into the ER lumen as translation continues.
No signal sequence.
If there's no ER signal sequence, translation finishes on that free ribosome in the cytosol. That protein is then destined for the cytosol itself, or it might have other targeting signals to direct it to the nucleus, mitochondria, or peroxisomes.
The source material also mentions manipulating this process using siRNA.
Ah, yes, small interfering RNA, siRNA. This is a powerful research tool and potentially a therapeutic one. siRNAs are short pieces of double-stranded RNA designed to match a specific target mRNA sequence.
And they shut down protein production.
They do. Once inside the cell, the siRNA gets incorporated into a complex called RISC, RNA-induced silencing complex. RISC uses the siRNA strand to find and bind to the complementary target mRNA. Once bound, it usually triggers the mRNA to be cut up and destroyed, or sometimes it just blocks the ribosome from translating it. Either way, production of that specific protein is effectively silenced.
Huge potential for targeting disease-causing proteins.
Immense. It's a major focus of drug development.
Okay, so proteins made in the RER need further processing and sorting.
Right.
Next stop, the Golgi apparatus.
The Golgi complex, yes. Think of it as the cell's post office or finishing school. It's typically located near the ER and the nucleus, especially prominent in cells that secrete a lot, like plasma cells making antibodies.
Structurally, it's a stack of flattened sacs.
Right, a stack of flattened, membrane-bound sacs called cisternae. It has a distinct polarity. Proteins and lipids arrive from the ER, usually in transport vesicles, at the cis face, the entry or forming face.
Then move through the stack.
They transit through the medial cisternae, undergoing sequential modifications, and finally reach the trans face, the exit or condensing face, where they are sorted and packaged into new vesicles for their final destinations.
What holds this stack together?
Golgins. Yes. Golgins are long, filamentous proteins that act like tethers, helping to maintain the Golgi's stacked structure and also helping to capture incoming vesicles from the ER and outgoing vesicles at the trans face.
And surrounding the main stack are these network regions.
Crucial sorting stations. The cis-Golgi network, CGN, right at the entry phase, does some initial sorting and, importantly, sends escaped ER-resident proteins back to the ER. The trans-Golgi network, TGN, at the exit phase, is the major sorting hub. This is where the final decisions are made about where a protein is going.
What kinds of modifications happen within the Golgi stacks? It's not just sorting, right?
No, it's major processing, especially for the carbohydrate chains, oligosaccharides, that were added in the RER. The Golgi enzymes trim and add sugars in a very specific step-by-step manner as the protein moves through the cisternae. This includes things like removing mannose sugars, adding sialic acid. Complex N-linked and O-linked glycosylation happens here.
And other chemical tags.
Yes, things like sulfation, adding sulfate groups, and phosphorylation, adding phosphate groups, famously used to target enzymes to the lysosome, also occur in the Golgi. These modifications act like postal codes, helping the TGNs sort the proteins correctly.
Okay, let's follow those sorted packages. The exocytic pathway is sending things out.
Right. Exocytosis is how cells release material. Vesicles budding from the TGN travel to the plasma membrane, fuse with it, and dump their contents outside. There are two main modes.
Constitutive and regulated.
Exactly. Constitutive secretion is the default pathway, happening continuously without a specific trigger. This is used for releasing components of the extracellular matrix, or plasma proteins from liver cells, for example. It just keeps going. Regulated secretion, on the other hand, is used for molecules that need to be released on demand, like hormones, neurotransmitters, or digestive enzymes. These products are stored in high concentrations in secretory granules near the plasma membrane. They only fuse and release their contents when the cell receives a specific signal, like a nerve impulse or a hormone binding to a receptor.
And polarity matters here too, right, where things get secreted.
Hugely important in polarized cells like epithelial cells. An exocrine gland cell secretes digestive enzymes apically into the duct. A liver cell secretes bile components apically, but plasma proteins basolaterally into the bloodstream. The TGN has to sort these vesicles to the correct membrane domain.
Okay, that's traffic out. What about traffic in? The endocytic pathway.
Endocytosis is the process of bringing material into the cell by engulfing it with the plasma membrane, forming an endocytic vesicle. This vesicle then travels inwards.
Where does it go?
Usually it first fuses with an early endosome, which is a sorting compartment just beneath the plasma membrane. From there, material can be recycled back to the plasma membrane, or it can progress to late endosomes, which are more acidic and are precursors to lysosomes. Eventually, late endosomes often mature into or fuse with lysosomes for degradation.
What are the different ways cells take things in?
The most specific way is receptor-mediated endocytosis. Cells have receptors on their surface for specific molecules, ligands, like LDL cholesterol or iron-carrying transferrin. When the ligand binds, the receptors cluster together, often in specialized regions called clathrin-coated pits. These pits invaginate and pinch off to form clathrin-coated vesicles containing the receptor-ligand complex, ensuring efficient uptake of specific cargo.
And caveoli.
Caveoli are another type of invagination, often seen in endothelial cells lining blood vessels and in smooth muscle cells. They're involved in a process called transcytosis, moving material across the cell, and also in certain signaling events. They have a different coat protein called caveolin.
Much of this incoming material and cellular waste ends up at the lysosome, the cell's recycling center or stomach.
That's a good analogy. Lysosomes are membrane-bound sacs, ranging in size, filled with a potent cocktail of hydrolytic enzymes, acid hydrolases, proteases, lipases, nucleases, glycosidases, about 60 different types.
And the acid part is key.
Absolutely crucial. These enzymes only work effectively at a very low pH, around 5.0. The lysosomal membrane contains proton pumps, ATPases, that actively pump H+ ions into the lysosome to maintain this acidity. The cytosol, being neutral, pH 7.2, provides a safety mechanism if a lysosome breaks. The enzymes are largely inactivated.
How does stuff get delivered to the lysosome for digestion?
Several routes. A primary lysosome, one that hasn't started digesting yet, can fuse with a late endosome carrying cargo from endocytosis. It can fuse with a phagosome containing something large engulfed by phagocytosis, like a bacterium. Or it can fuse with an autophagosome, which contains old organelles or bits of cytoplasm the cell wants to recycle.
Forming a secondary lysosome.
Right. The fusion product is the secondary lysosome, or endolysosome, where the actual digestion takes place thanks to the active hydrolases at low pH.
What happens to the leftovers? The indigestible bits.
They remain within the lysosome, which then becomes a residual body. In long-lived cells like neurons or heart muscle cells, these residual bodies can accumulate over time. They often contain oxidized lipids and proteins, forming aggregates known as lipofuscin, sometimes called the age pigment because it increases with age.
And if the lysosomal enzymes themselves are faulty?
That leads to lysosomal storage disorders, LSDs. There are over 50 different types, each caused by a defect in a specific lysosomal hydrolase, or sometimes in proteins needed for lysosomal function, like transport or acidification.
So the substrate that enzyme normally breaks down just builds up.
Exactly. It accumulates inside the lysosomes, causing them to swell, and eventually disrupting normal cell function. Tay-Sachs disease is a devastating example. A defect in beta-hexosaminidase, leads to buildup of GM2 ganglioside, primarily in neurons, causing progressive neurodegeneration. Danon disease involves a faulty LMP2 protein needed for autophagosome-lysosome fusion, leading to cardiomyopathy. They highlight how critical this waste disposal system is.
Okay, moving to another small but vital organelle, Peroxisomes. Different function entirely.
Very different. Peroxisomes are small, 0.2 to 1 presion, single-membrane organelles found in virtually all eukaryotic cells. They contain over 50 different enzymes involved in a variety of metabolic reactions.
Key roles.
Two stand out. First, they play a crucial role in beta oxidation, the breakdown of fatty acids. Specifically, they are essential for breaking down very long-chain fatty acids, VLCFAs, those longer than 22 carbons. Mitochondria handle the shorter ones.
And the other major role involves hydrogen peroxide.
Yes. Managing hydrogen peroxide. Many peroxisomal enzymes are oxidases, which produce TEX-2 as a byproduct during their reactions, since TEX2 is highly reactive and potentially damaging.
The peroxisome cleans up its own mess.
Instantly. Peroxisomes contain huge amounts of the enzyme catalase, which efficiently breaks down TEX-2 into harmless water and oxygen. This compartmentalization is key. Generate the toxic intermediate and destroy it in the same place.
And the proteins inside the peroxisome, they're made in the cytosol.
Yes, unlike proteins entering the ER pathway, peroxisomal matrix proteins are fully synthesized on free ribosomes in the cytosol and then imported into the peroxisome post-translationally. This requires special targeting signals and import machinery involving proteins called peroxins.
So defects in peroxins cause disease.
Severe ones. Mutations in peroxin genes disrupting protein import cause Zellweger syndrome, a devastating multisystem disorder usually fatal in infancy. And defects, specifically in the VLCFA beta oxidation pathway, cause conditions like X-linked adrenoleukodystrophy, XALD, leading to neurological damage and adrenal insufficiency.
Okay, last major organelle in this section, the powerhouse, mitochondria.
The energy factories, found in large numbers, especially in cells with high energy demands like muscle or neurons, typically at 0.5 to 0.0 nanomolar in size.
Classic double membrane structure.
Right. An outer membrane, which is quite permeable due to channel proteins called porins. Then an inner membrane, which is much less permeable, highly folded into structures called cristae.
Why the folding?
Sorry again. The inner membrane is where the electron transport chain and ATP synthase enzymes are located, the machinery for oxidative phosphorylation. The extensive folding of the cristae massively increases the surface area available for these ATP-producing reactions. The inner membrane is also uniquely rich in a phospholipid called cardiolipin, which helps make it impermeable.
And the space.
The space between the two membranes is the intermembrane space. The innermost compartment, enclosed by the inner membrane, is the matrix. The matrix contains the enzymes for the citric acid cycle, or Krebs cycle, and also mitochondrial DNA and ribosomes.
The inner membrane space has a key role in cell death signaling.
Yes, it contains cytochrome C. While essential for the electron transport chain within the mitochondrion, if it gets released out into the cytosol, usually due to cellular stress or damage, it acts as a potent trigger for activating the apoptotic pathway, initiating programmed cell death.
And the genetics are unique here. Mitochondria have their own DNA.
They do, a small circular chromosome called mtDNA. It encodes a handful of essential proteins for the respiratory chain, at least 13, plus mitochondrial tRNAs and rRNAs. But most mitochondrial proteins are actually encoded by the nuclear DNA, synthesized in the cytosol and imported.
Inheritance is strictly maternal.
Yes, you inherit your mitochondria, and thus your mtDNA, exclusively from your mother via the egg cell. Sperm mitochondria typically don't enter the egg or are destroyed shortly after fertilization. This maternal inheritance has important implications for genetic diseases caused by mtDNA mutations.
Which leads to the cutting-edge therapies, MRT.
Mitochondrial replacement therapies. These are advanced IVF techniques designed to prevent the transmission of severe mtDNA diseases from mother to child. The basic idea is to combine the nuclear DNA from the parents with healthy mitochondria from a donor egg.
How is it done? Two main ways.
Two main approaches currently. Pronuclear transfer, PNT, happens after fertilization. You take the fertilized egg from the mother with faulty mitochondria and another fertilized egg from a donor with healthy mitochondria. You remove the ponuclei, containing the parental nuclear DNA from both eggs. Then you transfer the parent's pronuclei into the enucleated donor egg. You end up with an embryo with nuclear DNA from the parents and mitochondria from the donor.
And the other way.
Maternal spindle transfer, MST, happens before fertilization. You take the mother's unfertilized egg and a donor's unfertilized egg. You remove the chromosome spindle, containing the mother's nuclear DNA, from her egg and transfer it into the enucleated donor egg. This reconstructed egg, with the mother's nuclear DNA and donor mitochondria, is then fertilized with the father's sperm.
Both aiming for the same result. Healthy mitochondria with parental nuclear DNA.
Exactly. It's ethically complex, but offers hope for families affected by devastating mitochondrial diseases.
Incredible technology. Okay, that covers the organelles. Let's move to the cell's internal framework, section 4, the cytoskeleton.
The cell skeleton, but also its muscles and highways. It's dynamic, not static like our bones. It provides mechanical strength, determines cell shape and polarity, allows cells to move and change shape, anchors organelles, and forms tracks for intracellular transport.
3 main types of filaments, differing in size and composition.
Right. We have microfilaments, the thinnest, about 7 millimeter diameter, made of the protein actin. Then intermediate filaments, around 10 millimeter, a diverse family of proteins providing tensile strength. And the largest are microtubules, about 25 millimeter diameter, hollow tubes made of the protein tubulin.
Let's start with actin filaments, or microfilaments, ubiquitous and dynamic.
Extremely dynamic. They're built from globular actin monomers, G-actin polymerizing, into a filament, F-actin. This requires ATP. Crucially, the F-actin filament has polarity.
A plus end and a minus end.
Yes. Monomers add preferentially to the plus end, also called the barbed end, while they tend to dissociate from the minus end, or pointed end. This polarity is the basis for directed growth and for actin's interaction with motor proteins like myosin.
And this leads to treadmilling.
Exactly. Under certain conditions, you can have simultaneous addition at the plus end and removal at the minus end. The filament stays roughly the same length, but the individual subunits are constantly flowing through it, like treads on a tank or escalator. This treadmilling is critical for things like cell crawling, where actin polymerization pushes the leading edge forward.
But actin doesn't act alone. Its function is totally dependent on a huge array of actin-binding proteins, ABPs.
Absolutely. These ABPs control actin polymerization, length, organization, and connections. They're the directors of the actin show.
Examples. How do they organize actin?
For forming parallel bundles, like the core of microvilli or filopodia, you have proteins like fibrin and vilin. For creating contractile bundles where actin filaments need to slide past each other, powered by myosin, you have proteins like alpha-actinin spacing them out appropriately, and myosin II itself acting as the motor.
What about networks instead of bundles?
Proteins like filamin cross-link actin filaments at wider angles, forming looser, gel-like networks, important for the cell cortex just under the plasma membrane. And for creating the highly branched networks that drive membrane protrusion in lamellipodia or during phagocytosis, the ARP23 complex is key. It nucleates new actin branches off the sides of existing filaments, typically at a 70-degree angle.
And proteins that control the monomers and filament growth.
Lots of those, too. Profilin promotes polymerization by helping to load G-actin onto the plus end. Thymosin beta-4 sequesters G-actin monomers, keeping a pool in reserve and preventing spontaneous polymerization. Gelsilin can sever existing filaments and cap the new plus ends, helping to break down networks quickly.
And finally, anchoring actin to other structures.
Crucial for stability and force transmission. Spectrin-based networks, using proteins like spectrin, fodrin, dystrophin, link actin to the plasma membrane itself. And at sites where the cell attaches to the extracellular matrix, called focal adhesions, actin filaments are linked via proteins like talin, vinculin, and kindlins to integrins, which are the transmembrane receptors binding the matrix outside.
Okay, moving up in size to microtubules, MTs. The rigid girders and highways.
Right. Hollow tubes, 25 millimeter diameter, built from dimers of alpha-tubulin and beta-tubulin. These dimers bind GTP. Like actin, microtubules also have polarity. The end with exposed beta-tubulin is the plus end, which is typically the faster-growing end. The alpha-tubulin end is the minus end.
But their dynamics are different. Not treadmilling, but dynamic instability.
Very different. Dynamic instability is the hallmark of microtubule behavior. Individual microtubules can switch abruptly and randomly between phases of slow growth and rapid shrinkage, called catastrophe. This depends on whether the tubulin dimers at the plus end have GTP or GDP bound. GTP-tubulin favors polymerization and stabilizes the tip, GTP intake cap, while hydrolysis to GDP-tubulin destabilizes it, leading to rapid depolymerization.
So they're constantly exploring the cell space.
Exactly, growing, shrinking, searching. This allows the cytoskeleton to reorganize rapidly. Microtubules don't just grow spontaneously, though. They usually nucleate from specific sites.
The MTOCs?
Microtubule organizing centers. The main one in most animal cells is the centrosome, located near the nucleus. It contains centrioles surrounded by pericentriolar material, which is rich in gamma-tubulin. This gamma-tubulin forms ring complexes that act as templates to nucleate microtubule growth, typically anchoring the minus ends. Basal bodies at the base of cilia and flagella are another type of MTOC.
And like actin, microtubules have associated proteins, MAPs.
Yes, microtubule-associated proteins. Some are structural MAPs like MAP1, MAP2, and tau. They bind along the sides of microtubules, stabilizing them and regulating spacing. MAP2 is mainly in dendrites, while tau is primarily in axons.
Tau is implicated in neurodegenerative diseases.
Heavily. In Alzheimer's disease and other tauopathies, tau becomes abnormally phosphorylated and forms aggregates, neurofibrillary tangles, inside neurons, disrupting transport and contributing to cell death.
Which brings us to the other class of MAPs, the motor proteins.
The transport machinery. These proteins use ATP hydrolysis to walk along microtubules carrying cargo like organelles and vesicles. Two main families, kinesins and dinins.
Directional movement.
Yes. Most kinesins are +N directed motors. They typically move cargo away from the cell center towards the periphery, like transporting neurotransmitter vesicles down the axon. This is anterograde transport. Dinines, specifically cytoplasmic dynein, are -N directed motors. They move cargo towards the cell center, recycling components back from the axon terminal, for example. This is retrograde transport. Dynines, specifically axonal dynein, are also the motors that power the bending of cilia and flagella.
Okay, the third component, intermediate filaments,
Yeah.
IFs, the ropes.
Good analogy. They are about 10 millimeter thick, rope-like filaments that provide tensile strength resistance to stretching and mechanical stress. Unlike actin and microtubules, they are not polar, they don't bind nucleotides, ATP, GTP, and they don't act as tracks for motor proteins. Their role is primarily structural integrity.
How are they assembled?
It's a hierarchical process. Two IF protein monomers coil around each other to form a dimer. Two dimers then associate in an anti-parallel staggered fashion to form a tetramer. This tetramer is the basic soluble subunit. Tetramers then associate end-to-end and laterally to form unit-length filaments, ULFs, and these anneal longitudinally into the final, long, tough filament. Because the tetramer is anti-parallel, the final filament lacks overall polarity.
And they're a diverse family, classified by type.
Very diverse, and their expression is often cell-type specific, which is useful in cancer diagnosis, identifying tissue of origin.
Types I and II.
Type I acidic and type 2 basic neutral keratins. These are characteristic of epithelial cells, skin linings. They always form heterodimers, one type I plus one type 2. Defects here cause skin blistering diseases like epidermolysis bullosa simplex, caused by mutations in keratin 5 or 14, making skin cells fragile.
Type 3.
Includes vimentin, found in mesenchymal cells like fibroblasts, desmin in muscle cells linking myofibrils, and GFAP, glial fibrillary acidic protein in astrocytes and other glial cells.
Type 4.
Neurofilaments. These are the main structural IFs in neurons, particularly abundant in axons, where they help determine axon diameter, which influences nerve conduction velocity. Abnormal accumulations are seen in diseases like ALS.
And type V, crucial for the nucleus.
Type V nuclear lamins, lamins ABC. These form the nuclear lamina, a meshwork lining the inner surface of the nuclear envelope, providing structural support to the nucleus and organizing chromatin. Mutations in lamin AC cause a wide range of diseases called laminopathies, including certain forms of muscular dystrophy, cardiomyopathy, and premature aging syndromes like progeria. It highlights how nuclear mechanics are vital for cell health.
Wow, that covers the entire physical structure. Let's shift to how cells talk to each other. Section V, cellular communication and signaling.
Essential for coordinating tissues and organism function, signaling happens over different distances.
Long range.
Two main types. Endocrine signaling. Hormones are secreted into the bloodstream and travel throughout the body to act on target cells far away. Think insulin or adrenaline. And neuronal signaling. Nerve cells transmit signals electrically along their axons and release chemical neurotransmitters at synapses to target specific cells very quickly, even over long distances.
And local chatter.
Several forms. Paracrine signaling. A cell releases local mediators that diffuse through the extracellular fluid and act on nearby different types of cells. Important in development and inflammation. Autocrine signaling. A cell releases signals that act on itself, often used for positive feedback or reinforcing cell identity.
And direct contact?
Contact-dependent signaling, or juxtacrine. Here, a signal molecule displayed on the surface of one cell binds directly to a receptor on an adjacent cell. No secreted molecule involved. Crucial during development for cell fate decisions and in the immune system for T-cell activation.
The type of signal molecule dictates where the receptor is located, right?
Generally, yes. Most signaling molecules, proteins, peptides, neurotransmitters like acetylcholine, are hydrophilic. They can't cross the plasma membrane, so they bind to plasma membrane receptors on the target cell surface, triggering a signal cascade inside.
But some signals can get inside.
Yes, small hydrophobic signaling molecules like steroid hormones, cortisol, estrogen, thyroid hormones, vitamin D, retinoids can diffuse directly across the plasma membrane. They typically bind to intracellular receptors, which are often located in the cytoplasm or the nucleus. These activated receptors usually act directly as transcription factors.
And the unusual case, nitric oxide.
Nitric oxide, NO, is a gas. It's small enough to diffuse rapidly across membranes. It acts locally by directly binding to and activating an intracellular enzyme, guanylate cyclase, which produces cyclic GMP, cGMP. No receptor protein needed in the conventional sense. Crucial for smooth muscle relaxation like dilating blood vessels.
The receptors themselves fall into major families: ion channel-linked, enzyme-linked, and the huge GPCR family.
Right. Ion channel-linked receptors, like ligand-gated ion channels, are simple. Ligand binding opens or closes the channel. Enzyme-linked receptors either have intrinsic enzyme activity, like receptor tyrosine kinases, or are directly associated with an enzyme that gets activated upon ligand binding. But the largest family by far is the G-protein coupled receptors, GPCRs. Thousands of different types in humans. They sense everything from light, rhodopsin, and smells to hormones and neurotransmitters. Most drugs target GPCRs or their downstream pathways.
They all have that characteristic 7-pass structure.
Yes, they snake back and forth across the membrane 7 times. Ligand binding on the outside causes a conformational change on the inside, allowing the GPCR to activate a heterotrimeric G protein, guanine nucleotide binding protein, associated with the interface of the membrane. This activated G protein then relays the signal onwards.
Let's look at some major signaling pathway mechanisms that use these receptors. The CAMP pathway is a classic.
Absolutely classic second messenger pathway. Ligand binds GPCR. GPCR activates a specific G protein, G stands for stimulatory. Activated G stimulates the enzyme adenyl cyclase. Adenyl cyclase converts ATP into cyclic AMP.
CAMP is the second messenger. What does it do?
CAMP diffuses through the cytosol and binds to the regulatory subunits of protein kinase A, pKa. This binding releases the active catalytic subunits of pKa.
And pKa phosphorylates things.
Exactly. pKa is a serine ethylene kinase. Its activated subunits can phosphorylate various target proteins in the cytosol, changing their activity. Importantly, they can also travel into the nucleus and phosphorylate transcription factors like CREB, CAMP response element binding protein. Phosphorylated CREB then binds to specific DNA sequences, CREs, and regulates the transcription of target genes. It's a pathway that allows for signal amplification and changes in gene expression.
Okay, next pathway, JAK-STAT, often used by cytokines.
Yes, cytokines and some hormones like growth hormone and erythropoietin, EPO, use this pathway. It's much more direct than the CAMP pathway.
How does it work?
The receptors themselves don't usually have intrinsic kinase activity, but they are associated with cytoplasmic tyrosine kinases called JAKs, Janus perneases. Ligand binding brings the receptor chains together, activating the associated JAKs.
JAKs phosphorylate each other and the receptor.
Right. This creates docking sites on the receptor for STAT proteins, signal transducers, and activators of transcription. STATs have SH2 domains that recognize these phosphatyrosine sites. Once docked, the JAKs phosphorylate the STATs.
Then what happens to the STATs?
Phosphorylated STATs dissociate from the receptor, dimerize, pair up, and the STAT dimer translocates directly into the nucleus. There, it binds to specific DNA sequences and activates the transcription of target genes. For EPO signaling, this leads to the production of proteins needed for red blood cell development. Very direct route from receptor to gene expression.
Now for some key developmental pathways. The hedgehog H pathway.
Crucial for embryonic patterning, limb development, neural tube formation. HH signaling involves a receptor called patched, PTCH1, and another transmembrane protein called smoothened, SMO.
How do they interact?
In the absence of a hedgehog ligand, PTCH1 inhibits SMO activity. When H ligand binds to PTCH1, this inhibition is relieved and SMO becomes active. Active SMO then initiates a cascade that ultimately controls the activity of the green family of transcription factors.
GLI proteins can be activators or repressors.
Yes, that's a key complexity. Depending on the context and processing, green proteins, GLI1, GL2, GL3, can either activate or repress target gene transcription. Misregulation of the HH pathway is implicated in several cancers, like basal cell carcinoma, medulloblastoma, and developmental disorders like Gorlin syndrome.
Another major player in development, the Wnt pathway.
Equally fundamental, involved in body axis formation, cell fate specification, stem cell maintenance. Wnt ligands bind to receptors of the frizzled FCD family, often requiring co-receptors like LRP56.
And the key downstream player is beta-catenin.
Yes, in the canonical Wnt pathway. In the absence of Wnt signal, beta-catenin in the cytoplasm is constantly being targeted for degradation by a destruction complex involving proteins like APC and GSK3. When Wnt binds its receptors, this destruction complex is inactivated.
So beta-catenin levels rise.
Exactly. Stable beta-catenin accumulates, translocates into the nucleus, and partners with transcription factors of the TCFLEF family to activate the expression of Wnt target genes. This pathway is also heavily regulated by other factors like arspondins, ARSPO, and E3 ubiquitin ligases that control receptor levels. Its dysregulation is strongly linked to colorectal cancer and other malignancies, but it's also a major target for regenerative medicine approaches.
Finally, the TGF-beta-beta-P pathway, also involved in development, but with that tricky dual role in cancer.
Right. This family includes transforming growth factor beta, TGF-beta itself, bone morphogenetic proteins, BMPs, activins, and others. They all bind to receptor complexes composed of type I and type II serine etherinine kinases.
Ligand binding brings the receptors together.
Yes, the ligand usually binds the type II receptor, which then recruits and phosphorylates the type I receptor, activating its kinase domain. The activated type I receptor then phosphorylates downstream signaling proteins called SM-betals.
Different SMADs for different signals.
Yes. TGF-beta-beta and activins typically activate SMAD2 and SMAD3. BMPs activate SMAD1, SMAD, and SMAD8. These receptor-regulated SMADs then form a complex with a common partner, SMAD4.
And this complex goes to the nucleus.
The RSMAD-SMAD4 complex translocates to the nucleus, where it partners with other transcription factors to regulate target gene expression. The effects are highly context-dependent. As you said, TGF-beta can inhibit proliferation in early-stage cancers, acting as a tumor suppressor. But in later stages, cancer cells often hijack the pathway, and TGF-beta promotes invasion, metastasis, and immune suppression.
That covers the major communication lines.
Yeah.
Let's turn to the control center itself. Section 6, the nucleus, cell cycle, and cell fate.
The nucleus repository of the genetic blueprint, DNA. It's enclosed by the nuclear envelope.
Which is a double membrane.
Yes, two concentric membranes. The inner nuclear membrane, INM, and the outer nuclear membrane, ONM. The ONM is continuous with the membrane of the endoplasmic reticulum and often has ribosomes attached. The space between the INM and ONM is continuous with the ER lumen.
And the structure is supported internally.
By the nuclear lamina, that meshwork of lamin intermediate filaments we discussed earlier underlying the INM. It provides mechanical support and helps organize chromatin.
How does anything get in or out? The nuclear pore complexes, NPCs.
These are huge, intricate protein structures, thousands of them embedded in the nuclear envelope where the INM and ONM fuse. They act as highly selective gates regulating all traffic between the nucleus and the cytoplasm.
Selective and directional.
Absolutely. Small molecules, 40 kilodia, can diffuse through passively, but larger molecules, especially proteins and RNA, require active regulated transport. Proteins destined for the nucleus must have a specific tag, a nuclear localization sequence, NLS.
Recognized by import proteins.
Yes, recognized by importance, a type of karyophorine, which then chaperone the cargo protein through the NPC into the nucleus. Export works similarly, with exportins recognizing nuclear export signals. NES. The whole process is coordinated and powered by the RAN-GT pace cycle, which essentially dictates directionality by maintaining a high concentration of RAN-GTP inside the nucleus and RAN-GDP outside.
Inside the nucleus, the DNA isn't just floating around. Chromatin organization.
Right. Chromatin is the complex of DNA and associated proteins, mostly histones, but also non-histone proteins. The sheer amount of DNA, 2 meters in a human cell, has to be compacted incredibly efficiently to fit inside the nucleus, typically 10 another diameter.
The first level of packing involves histones.
Yeah, the fundamental unit is the nucleosome. DNA wraps about 1.7 times around a core particle consisting of 8 histone proteins, 2 each of H2A, H2B, H3, and H4, the histone octamer. This looks like beads on a string under the electron microscope, forming the 10-millimeter chromatin fiber.
And it gets packed further.
The nucleosomes then coil up, often involving another histone, H1, the linker histone, to form the 30-nanometer fiber. This fiber is then organized into larger loops, which are anchored to a nuclear scaffold or matrix. The degree of compaction varies.
leading to euchromatin and heterochromatin.
Exactly. Euchromatin is the less condensed form, generally corresponding to regions where genes are actively being transcribed, or are poised to be. It appears pale or dispersed in electron micrographs. Heterochromatin is highly condensed, transcriptionally inactive chromatin. It often appears dark and clumped, frequently located near the nuclear envelope or around the nucleolus. The inactive X chromosome in female mammals, the bar body, is a classic example of facultative heterochromatin.
The DNA itself is organized into linear units, chromosomes, defined by centromeres and telomeres.
Right. Each chromosome is a single, extremely long DNA molecule complexed with proteins. The centromere is a constricted region, essential for proper segregation during cell division. It's where the kinetochore, the protein complex that attaches to microtubules, assembles. Telomeres are the specialized structures at the physical ends of the linear chromosomes.
Protective caps.
Protective caps, exactly. In humans, they consist of thousands of repeats of the sequence, TTGG. They prevent the ends from being recognized as DNA breaks and protect against degradation. They also solve the end replication problem, the fact that standard DNA polymerase can't fully copy the very end of a linear molecule.
But they shorten over time.
In most somatic cells, yes. With each round of replication, a little bit of the telomere sequence is lost. Eventually, they become critically short, which signals the cell to stop dividing a state called replicative senescence. This telomere shortening is considered a biological clock linked to cellular aging and potentially organismal aging.
Unless the cell has telomerase.
Right. Telomerase is a specialized enzyme, a reverse transcriptase, that can add TTG repeats back onto the telomere ends, counteracting the shortening. It's active in germ cells, allowing inheritance of full-length chromosomes, stem cells, allowing extensive proliferation, but usually inactive in most differentiated somatic cells. Cancer cells very often reactivate telomerase, allowing them to bypass senescence and achieve replicative immortality.
Okay, let's look at the process of cell division itself, the cell cycle G1, S, G2, M.
The orderly sequence of events by which a cell duplicates its contents and divides in two. The main phases are interphase, the period of growth and DNA replication, and M phase, mitosis, nuclear division, and cytokinesis, cytoplasmic division.
Interphase is subdivided.
Into G1, S, and G2. G1 phase, gap 1, is a period of growth and metabolic activity where the cell prepares for DNA synthesis. S phase synthesis is when the cell replicates its entire genome. G2 phase, gap 2, is another growth period where the cell prepares for mitosis.
How is progression through these phases controlled? Cyclins and CD-Sheweets?
Yes, the cell cycle engine is driven by a family of protein kinases called cyclin-dependent kinases, CDKs. CDKs are inactive on their own. They need to bind to regulatory proteins called cyclins, whose concentrations rise and fall in a cyclical pattern during the cell cycle.
Different cyclin-CDK pairs control different transitions.
Exactly. For example, CDK4/6 pairs with cyclin D to drive progression through G1. CDK2 pairs with cyclin E to initiate S phase, and then with cyclin A to continue S phase. CDK1 pairs first with cyclin A and then cyclin B to drive entry into and progression through M phase. CDK1 cyclin B is often called MPF, maturation promoting factor.
And getting out of a phase often requires destroying the cyclin.
Crucial point. Transitions are made unidirectional and irreversible largely by the regulated proteolysis, destruction of cyclins, via the ubiquitin proteasome system. Specific E3 ubiquitin ligases like SCF and the anaphase-promoting complex cyclosome, APCC, target specific cyclins for ubiquitination at the right time, marking them for degradation by the proteasome. This inactivation of CDKs allows the cell to exit one phase and enter the next.
And there are safety mechanisms. Checkpoints.
Absolutely vital. Checkpoints are surveillance mechanisms that monitor the integrity of the genome and the proper progression of cell cycle events. They can halt the cycle if something is wrong, allowing time for repair. Key checkpoints occur late in G1, checking for DNA damage before S phase, during S phase, monitoring replication, and late in G2, checking for DNA damage and complete replication before M phase, as well as during M phase, the spindle assembly checkpoint, ensuring all chromosomes are properly attached before anaphase.
P53 plays a major role here.
The guardian of the genome, P53. It's a tumor suppressor protein that gets activated in response to DNA damage or other cellular stress. Activated P53 acts primarily as a transcription factor. One of its key targets is the gene encoding P21, which is a CDK inhibitor protein. P21 binds to and inhibits G1S CDKs, like CDK2 cyclin E and S CDKs, causing cell cycle arrest, typically in G1 or G2.
Giving time for repair or triggering apoptosis if damage is too severe.
Exactly. Loss of functional p53 is incredibly common in human cancers. Example, Li-Fraumeni syndrome is caused by inherited p53 mutations, as it removes this critical brake on proliferation, especially for cells with damaged DNA.
Okay, let's look at mitosis itself, the actual division for somatic cells.
M phase. It's a continuous process, but usually divided into stages for convenience. Prophase. Chromatin condenses into visible chromosomes, the nuclear envelope breaks down, and the mitotic spindle, made of microtubules, starts to form between the two centrosomes, which have moved to opposite poles of the cell.
Then metaphase.
Metaphase. The chromosomes, each consisting of two identical sister chromatids held together by cohesin proteins, align along the equatorial plate, the metaphase plate, of the spindle. This alignment is mediated by microtubules from opposite poles attaching to the kinetochore on each sister chromatid.
Anaphase, the separation.
Anaphase. This starts abruptly when the APCC triggers the degradation of securing, which releases an enzyme called separase. Separase cleaves the cohesin complexes holding the sister chromatids together. The sister chromatids, now considered individual chromosomes, are pulled apart towards opposite spindle poles by the shortening kinetochore microtubules.
Finally, telophase and cytokinesis.
Telophase. The separated chromosomes arrive at the poles and begin to decondense. Nuclear envelopes reform around the two sets of chromosomes, creating two new nuclei. Cytokinesis, which usually begins during late anaphase or telophase, is the physical division of the cytoplasm. In animal cells, this involves the formation of a contractile ring of actin and myosin filaments at the equator, which pinches the cell in two, forming a cleavage furrow.
Result: two genetically identical daughter cells. But for gametes, we need meiosis.
Right. Meiosis is the specialized cell division that produces haploid gametes, sperm and eggs, from diploid precursor cells. It involves two consecutive nuclear divisions, meiosis the first and meiosis the second, but only one round of DNA replication before meiosis the first.
Meiosis the first is the reductional division.
Yes, meiosis I separates homologous chromosomes. Its prophase I is particularly long and complex, subdivided into stages: leptotene, zygotene, pachytene, diplotene, diakinesis.
Key events in prophase I.
Zygotene. Homologous chromosomes pair up precisely along their entire length, a process called synapsis, mediated by the synaptonemal complex, a protein structure that holds them together. Pachytene. Synapsis is complete. Crucially, crossing over, genetic recombination, occurs between non-sister chromatids of the homologous pair, exchanging segments of DNA. This creates new combinations of alleles.
Where the crossovers happen become visible later.
Yes, in diplotene, the synaptonemal complex breaks down, and the homologous chromosomes start to separate, but they remain held together at the sites where crossing over occurred. These points of attachment are visible as chiasmata, singular, chiasma.
Then metaphase I and anaphase I.
In metaphase I, the paired homologous chromosomes, bivalents, held together by chiasmata, align on the metaphase plate. The orientation of each pair is random, independent assortment. In anaphase I, homologous chromosomes separate and move to opposite poles. Sister chromatids remain attached at their centromeres. This separation of homologs is the key reductional step.
So meiosis pore produces two haploid cells, but each chromosome still have two chromatids.
Exactly. Then, after a usually brief interphase with no S phase, the cells enter meiosis II. Meiosis II is very similar mechanically to mitosis. Sister chromatids separate during anaphase II. The final result is 4 genetically distinct haploid cells.
Errors in meiosis, like non-disjunction.
Disjunction. The failure of homologous chromosomes in meiosis I or sister chromatids in meiosis II to separate properly is a major cause of aneuploidy, abnormal chromosome number, in gametes. This leads to conditions like trisomy 21 Down syndrome, where individuals have 3 copies of chromosome 21. The frequency of non-disjunction increases significantly with maternal age.
Okay, that covers division. Let's finish with cell fate, particularly programmed cell death and degradation systems. Apoptosis versus necrosis.
2 ways cells die. Necrosis is messy, accidental cell death usually caused by injury, toxins, or lack of oxygen. The cell swells, membranes rupture, contents spill out, causing inflammation. Apoptosis is programmed cell death, an active, orderly process of cellular self-destruction.
Why is apoptosis needed?
Essential for normal development, for example, removing webbing between fingers, tissue homeostasis, balancing cell proliferation, and removing damaged, infected, or cancerous cells. It's clean, the cell shrinks, chromatin condenses, the nucleus fragments, and the cell breaks up into membrane-enclosed apoptotic bodies that are rapidly engulfed by phagocytic cells, preventing inflammation.
How is it triggered? 2 main pathways.
Yes. The extrinsic pathway is triggered by external signals. like Fas ligand binding to the Fas-Neth receptor on the cell surface. This recruits adapter proteins and initiator caspases, like caspase 8. The intrinsic pathway, or mitochondrial pathway, is triggered by internal stress, like DNA damage, growth factor withdrawal, or ER stress. This leads to changes in the mitochondrial outer membrane permeability, causing the release of cytochrome C into the cytosol.
And both pathways converge.
They both converge on the activation of executioner caspases, primarily caspase 3, nitative 6, and natica 7. Caspases are proteases, cysteine aspartic acid-specific proteases that exist as inactive procaspases. Once activated, often in a cascade, they cleave hundreds of target proteins throughout the cell, dismantling the cellular structure, degrading the nuclear lamina, activating DNases to chop up DNA, cleaving cytoskeletal proteins, leading to the characteristic apoptotic morphology.
Beyond wholesale destruction via apoptosis, the cell has ongoing degradation systems for maintenance, the ubiquitin protein pathway.
This is the primary system for degrading most short-lived regulatory proteins, like cyclins, and also misfolded or damaged proteins synthesized in the cytosol or ER, via ER. Proteins destined for degradation are tagged with a chain of ubiquitin molecules, usually by a cascade of enzymes ending in an E3 ubiquitin ligase, which provides specificity.
And a tag targets them to the cardisome.
The polyubiquitin tag is recognized by the 26S proteasome, a large barrel-shaped protein complex. The proteasome unfolds the tagged protein, threads it into its central chamber, and chops it up into small peptides, releasing the ubiquitin for reuse. It's like a molecular shredder.
And for bigger things, like whole organelles, autophagy.
Autophagy, self-eating, is the process for degrading bulk cytoplasm, long-lived proteins, protein aggregates, and even entire organelles like damaged mitochondria or peroxisomes. A double membrane, the phagophore likely originating from ER or Golgi, engulfs the targeted material, forming a vesicle called the apophagosome.
Which then fuses.
The autophagosome fuses with a lysosome, forming an autolysosome. The lysosomal hydrolyses then degrade the contents. Autophagy is crucial for nutrient recycling during starvation, clearing damaged components, and protecting against neurodegeneration and infection.
And the specific autophagy of mitochondria is mitophagy.
Yes, the targeted removal of damaged or superfluous mitochondria. A key pathway involves the protein kinase, PINK1, and the E3 ubiquitin ligase, parkin. When a mitochondrion is damaged, VAG loses its membrane potential, PINK1 accumulates on the outer membrane and recruits parkin from the cytosol.
Parkin tags the mitochondrion.
Parkin ubiquitinates proteins on the mitochondrial outer surface. This ubiquitin coat serves as a signal to recruit the autophagy machinery, leading to the engulfment of the damaged mitochondrion by an autophagosome and its subsequent degradation in lysosomes. Defective mitophagy, often due to mutations in PINK1 or parkin, is strongly linked to early-onset Parkinson's disease, highlighting the importance of mitochondrial quality control.
We've covered the internal machinery. Let's conclude with Section 7, cell polarity and intercellular junctions. How cells organize themselves into tissues.
Especially important for epithelial cells, which form barriers. They exhibit distinct polarity with an apical surface facing the outside world, or lumen, and a basolateral surface facing the underlying tissue and blood supply. This polarity is established and maintained by specialized structures and junctions.
The apical surface often has specializations, cilia and microvilli.
Right. Cilia are hair-like projections. Motile cilia, like in the respiratory tract or fallopian tubes, have a characteristic 9+2 arrangement of microtubules in their core, the axoneme, and contain dyning motor arms that generate movement to propel fluid or mucus. Flagella, like on sperm, have a similar structure and function for cell locomotion. A primary cilia, on the other hand, are usually solitary, non-motile, often lacking the central pair of microtubules, a 9+0 structure, and function as sensory antennae, detecting chemical or mechanical signals. They play critical roles in many signaling pathways, including hedgehog.
Building and maintaining these requires constant transport.
Yes, intraflagellar transport, IFT. It's a dedicated system using motor proteins, kinsin 2 moving cargo out towards the tip, anterograde, and cytoplasmic dynein 2 moving things back towards the base, retrograde, along the axonal microtubules. IFT is essential for assembling and maintaining cilia. Defects cause ciliopathies, a wide range of human diseases like Bardet-Biedl syndrome, polycystic kidney disease, Cardiganer syndrome.
And microvilli, more static.
Microvillis are shorter, non-motile, finger-like extensions designed purely to increase surface area for absorption, forming the brush border seen on intestinal and kidney tubule cells. Their core is made of stable bundles of actin filaments cross-linked by proteins like fibrin and vilin. These bundles are anchored into a network of actin and spectrin in the apical cytoplasm called the terminal web.
Now, the junctions holding cells together and creating seals, the apical junctional complex.
This is a belt-like series of junctions found near the apical surface of epithelial cells, crucial for polarity and barrier function. It typically consists of three components, from apical to basal: tight junctions, adherens junctions, and desmosomes, though desmosomes are often considered separate spot junctions.
The tight junctions, zonulae occludentes, are the most apical.
Yes. They form a continuous seal around the cell, completely obliterating the space between adjacent cell membranes at certain points. They are formed by transmembrane proteins like occludins and claudins linking across the gap, and intracellularly connecting to ZO, zonula occludens proteins, which link to the actin cytoskeleton.
Two key functions.
First, they create a paracellular permeability barrier, restricting the passage of water and solutes between the cells, forcing transport to go through the cells transcellularly. The tightness varies depending on the specific claudins expressed. Second, they act as an intramembrane diffusion barrier, a fence that prevents lipids and proteins in the apical membrane domain from diffusing laterally into the basolateral domain, thus maintaining cell polarity.
Just below the tight junctions.
The zonula adherens, or adherens junction, often forming a continuous belt, the adhesion belt. This junction provides strong cell adhesion, linking the actin cytoskeletons of adjacent cells.
Using cadherins?
Yes, the key transmembrane proteins here are classical cadherins, like E-cadherin in epithelia. Cadherins mediate calcium-dependent homophilic adhesion, meaning they bind to the same type of cadherin on the neighboring cell. Intracellularly, cadherins link via catenin proteins, alpha, beta, p120, to the actin cytoskeleton. The aphidinectin system also contributes to adherens junction formation and stability.
Let's quickly cover the main adhesion molecule families. Cadherins are one.
Adherents, calcium-dependent homophilic adhesion, crucial for tissue cohesion, includes classical cadherins, E, N, p-cadherin, and desmosomal cadherins, desmoglein, desmocolons.
Lectins.
Selectins, also calcium-dependent, but they are lectins, meaning they bind to specific carbohydrate structures on glycoproteins, glycolipids on other cells, heterophilic adhesion. Mediate transient weaker adhesions, famously involved in the initial rolling of white blood cells on the blood vessel endothelium during inflammation, LEP selectins. Integrins. Calcium-independent heterodimers and alpha and a beta subunit mediate both cell-cell and, very importantly, cell extracellular matrix, ECM adhesion. They typically bind ECM proteins like fibronectin, laminin, collagen outside the cell and link to the actin cytoskeleton inside the cell via adapter proteins like talin, vinculin, and kinlins at structures called focal adhesions or focal contacts. Integrins can signal bidirectionally, outside in and inside out signaling.
Okay, back to junctions. Desmosomes, maculae adherentes, spot welds.
Exactly. Strong, spot-like adhesive junctions, particularly abundant in tissues subject to mechanical stress like skin and heart muscle. They use specialized desmosomal cadherins, desmogleins and desmocolons, to link cells across the gap. The key difference from adherens junctions is that intracellularly, desmosomes anchor to intermediate filaments, keratins and epithelia, desmin and muscle, via dense cytoplasmic plaques containing proteins like plakoglobin and desmoplakin. This links the IF networks of adjacent cells, providing immense tensile strength.
And attaching the cell to the basement membrane. Hemenzymosome.
Asymmetric junctions found on the basal surface of epithelial cells, anchoring them firmly to the underlying basal lamina, part of the ECM. They use specific integrins, notably alpha-beta-4,4 as the transmembrane linkers, connecting to laminins in the basal lamina outside, and anchoring to the intermediate filament, keratin, cytoskeleton, inside via linker proteins like plectin. They look like half a desmosome, hence the name.
Finally, the communicating junctions, gap junctions.
These provide direct cytoplasmic continuity between adjacent cells. There are clusters of channels that span the gap between cells. Each channel, called a connexin, is formed by 6 protein subunits called connexins. 2 connexins, one from each cell, align end to end to form a continuous aqueous pore,
allowing small molecules to pass.
Yes, ions, second messengers like CMP and IP3, small metabolites, sugars, amino acids, up to about 1 kD can pass freely through gap junctions. This allows for direct electrical coupling, crucial in heart muscle, and metabolic cooperation between cells in a tissue.
We've done it from the outer boundary through the organelles, the cytoskeleton, the nucleus, signaling, division, death, And finally, the connections between cells, a true mastery tour.
It's an incredible amount of interconnected machinery. The synthesis, I think, is seeing the cell not just as a bag of parts, but as this highly dynamic, integrated system where structure, function, regulation, and communication are constantly interacting.
And the emphasis on dynamic equilibrium, quality control, ERD, checkpoints, autophagy.
Exactly. Multiple layers of quality control and tight regulation are built in everywhere, from protein folding in the ER to DNA replication checkpoints, controlled degradation via the proteasome and autophagy, and the precise orchestration of signaling pathways. It's all about maintaining stability and function in a complex environment.
So for our final provocative thought, let's circle back to those degradation systems. Autophagy, mitophagy, the proteasome, and their link to aging and disease.
Right. We touched on lipofuscin accumulation and the link between faulty mitophagy and Parkinson's. The bigger picture emerging is that the efficiency and fidelity of these cellular cleaning crews seem to be fundamentally linked to health span and lifespan.
A breakdown in waste disposal leads to cellular decline.
It appears so. As cells age, these systems may become less efficient. Damaged proteins, aggregates, and faulty organelles, especially mitochondria, which are major sources of damaging reactive oxygen species, can accumulate. This buildup contributes to cellular dysfunction, increased stress, and ultimately senescence or death.
So the failure of these basic housekeeping functions could be a central driver of aging and age-related diseases like Alzheimer's and Parkinson's.
That's a major hypothesis being actively investigated. It suggests a profound connection. The cell's ability to take out its own trash, particularly worn-out mitochondria, might be a key determinant of how long it functions properly and perhaps how long we live healthily.
Which raises the question, could we intervene? Could boosting these degradation pathways extend health span?
That's the multi-billion-dollar question. Understanding exactly how autophagy, mitophagy, and proteasome function decline with age, and finding ways to safely and effectively enhance their activity, perhaps through lifestyle changes, diet, or pharmacological interventions, is a huge frontier in biomedical research. It shifts the focus from just fixing damage to proactively maintaining cellular quality control. A fascinating place to leave our deep dive today.